Okay, so good morning, children. Yesterday we were talking about how the fatty acid synthase complex is working to synthesize a fatty acid, say a palmitic acid, right? So it involves the fatty acid synthase structure. It's a multi-enzyme complex made up of two different subunits, and the two subunits are joined together, or they are lying side by side, adjacent to each other in a head-to-tail fashion, so that the peripheral arm of one monomer is facing the central arm of the other monomer, right? And the E1, E2, E3 along with the peripheral arm forms the functional part of the E4, 5, 6, 7 with the central arm of the second monomer. So that is how the functional aspect of fatty acid synthase uh, resides, right? So there are, even though the structural arrangement is something different from that of the functional domain, the two molecules, the two monomers works parallelly simultaneously in such a way that it can synthesize two fatty acids at a time. So the substrate molecule for this fatty acid synthesis is the acetyl CoA, and again the reaction involves a condensation of the two carbon units in stepwise manner, and it is happening in a cycle, in a cyclic way, so that each cycle ends up with the formation of a fatty acid with two carbon greater, right? With the chain length will be along getting extended with two carbon at the end of each cycle, and the cycle ends up with the formation of a C16 fatty acid, and we call it as a palmitic acid, right? So two carbon acetyl CoA molecules enters into the fatty acid biosynthetic pathway through 8 cycles. 8 cycles, or it is happening in a stepwise manner, and enters through, it passes through eight cycles to end up in the formation of the fatty acid C16 fatty acid, the palmitic acid, right? So it is almost the reversal of the beta oxidation. In the beta oxidation, what is happening, the C16 palmitic acid is undergoing breakage, or it undergoes degradation in a cyclic manner, so that each cycle ends up with the release of two carbon, so that the chain length will be reduced with two carbon, reduced by two carbon atoms, right? So C16 palmitic acid, when enters or when ends up in the first round of beta oxidation, it becomes, the chain length will be reduced to C14, right? In the same way, the C16, the 16 carbon atoms will be undergoing breakage to give rise to release two acetyl CoA, the two carbon molecule, the acetyl CoA at the end of every cycle, right? As a result of which, eight molecules of acetyl CoA can be synthesized from one molecule of fatty acid when it enters into beta oxidation. Now, in case of fatty acid biosynthesis, the reverse happens. That is, the two carbon acetyl CoA molecules condenses together, so that the reaction involves the chain elongation, so that at the end of each cycle, the chain length will be increased by 2. Okay, so again, when two carbon acetyl CoA, it has to pass through 8 different cycles to end up in the formation of palmitic acid. Okay, and again, the pathway is happening in the cytosol, and it's carried out by a multi-enzyme complex. And before the fatty acid synthase works, there are two other stages to be happen. The acetyl CoA has to be first translocated from the mitochondria into the cytosol. This is followed by the carboxylation reaction of acetyl CoA, because the acetyl CoA molecule, when condenses, a large amount of energy is required, and it is a purely thermodynamically unfavorable phenomenon, because the two molecules are having equal energy. So it's not possible for them to simply come and get condensed. So what is happening? Out of the two acetyl CoA molecules, one of the acetyl CoA molecule will be remaining as such, while the other will be undergoing carboxylation reaction to give rise to malonyl CoA, right? So as a result of which, or at the end of the stage 2, or when it enters into the fatty acid, the stage 3, that is when the fatty acid synthase start working, this raw materials required for the condensation reaction will be one molecule of acetyl CoA, the two carbon molecule acetyl CoA, and the other molecule is coming in the form of malonyl CoA rather than acetyl CoA. Clear? So out of the 8 molecules of acetyl-CoA that is responsible for synthesizing one molecule of palmitic acid, only one of the acetyl-CoA will be remaining as such, while the remaining 7 acetyl-CoA molecules will be undergoing carboxylation reaction to give rise to malonyl-CoA. Right? So out of the 8 molecules of acetyl-CoA, only a single molecule of acetyl-CoA is remaining as such, or it enters the fatty acid synthesis in the form of acetyl-CoA, while the remaining 7 molecules of acetyl-CoA that are ready to get entered into the fatty acid biosynthesis will be coming in the form of malonyl CoA, right? So again, so that the stage that was the stage 1 and 2. That is, first one is the translocation, which is carried out by malate shuttle. The second one is the carboxylation reaction of acetyl-CoA, which is carried out by again a multi-enzyme complex, which is known as acetyl-CoA carboxylase, which is a very important enzyme participating in the fatty acid biosynthesis, right? Because it requires carboxylation, so carbon dioxide is required. Carbon dioxide is coming in the form of bicarbonate ions. The bicarbonate ions releases the carbon dioxide not directly to the acetyl-CoA, but it is first accepted by the cofactor biotin, right? Biotin, so that biotin is undergoing biotin carboxybiotin form, and this is happening in the presence of an enzyme biotin carboxylase. So biotin carboxylase converts biotin to carboxybiotin first, and this carboxybiotin then releases the carbon dioxide to the acetyl-CoA by means of carboxybiotin transferase, right? So there are 2 enzyme activities that are residing in the acetyl-CoA carboxylase enzyme. First one is a biotin carboxylase and the other one is carboxybiotin transferase, which finally converts acetyl-CoA to malonyl-CoA, right? And it again, it's an ATP requiring process. Now it enters into the stage 3. What is happening in stage 3? Stage 3 is the condensation reaction or the polymerization of acetyl-CoA molecules to give rise to fatty acyl-CoA, right? So the again, it is carried out by a multi-enzyme complex in which there are 7 enzyme activities residing in a single molecule of fatty acid synthase, and each of these 7 enzymes are functioning in an autonomous manner. That is, they're working in an independent manner, but the product will not be leaving the fatty acid synthase. Once it is formed, it is immediately focused or it is immediately channeled to the next enzyme activity, right? So, so that the final product only will be coming out of the fatty acid synthase complex. Only after the C16 palmitic acid is formed, then only it will be getting liberated out. Otherwise, at the end of each cycle, it will be taken to the next step. Without coming out of the protein molecule or the enzyme molecule, right? And as seven reactions are there out of the 7 enzymes, and not there are 7 enzymes participating. Out of this, the thioesterase enzyme, which is the last enzyme, it is coming into action only at the final cycle because it carries out a thiolytic cleavage. So if we are keeping that thioesterase apart, we will be focusing on the remaining 6 enzymes, that is E1, E2, E3, E4, E5 and E6, right? So thioesterase is having ACP acids, that is ACP is connected to thioesterase. So ACP is acting as the it is acting as a central thiol group, or it is having a pan-SH group. This pan-SH group is having the capacity, or it's having the power to swing throughout the molecule, so that each molecule, each product formed after the reactions will be taken from one enzyme to the other. Okay, so that is how the structure of the fatty acid synthesis is coming. And again, the first enzyme being the acetyl transacylase, and the second enzyme is, or E2 is malonyl transacylase. So what is the function of these 2 enzymes? These 2 enzymes are carrying out the priming reactions, because we have to collect the raw materials first and they have to be brought together. That is carried out by the first 2 steps, which is known as the priming reactions. So what does the priming reactions do? The first enzyme is the acetyl transacylase. So what does acetyl transacylase do? Acetyl transacylase takes up the acetyl group or acetyl moiety from the acetyl-CoA, so that the CoA will be liberated. Only the acetyl group is picked up by the enzyme, and this is transferred to the peripheral thiol group, that is the cis-SH group. Clear? So the if suppose we are the peripheral group is cis is the monomer 1. If this we are concentrating on the monomer E1, E2, E3 of the monomer 1, then the E1 of monomer 1 picks up the acetyl group from acetyl-CoA, and it will be picked. It will be located on the peripheral arm or the cis-SH arm. I'll show you the picture. Yeah. Yes. So this is the acetyl-CoA. So this is our fatty acid synthase complex, right? It's having the seven different enzyme activities, of which the acetyl transacylase and malonyl transacylase, right? And ketoacyl synthase. So these are the enzymes which belong to the monomer 1. Okay. So the acetyl transacylase picks up the acetyl group only, so that the CoA-SH will be released. CoA SCOA, right? So SCOA SCOA ഇവിടെ വരും, H എവിടുന്ന വരണമെന്നുള്ളത് I'll show you. So acetyl transacylase picks up the acetyl group only from the acetyl CoA, and it will be getting placed on the ketoacyl synthase E3, or E3 is having the peripheral arm or cis SH arm. അപ്പോൾ ഈ cis SH ന് ഒരു H ഉണ്ട്. So H is removed, and to that position the CH3CO group will be going and getting attached. അപ്പോൾ ആ H ആണ് ഇവിടേക്ക് വരുന്നത്. So that CoA-SH, SCOA is released in the form of CoA-SH. So H comes from this SH. SH le sulfhydryl group, ഈ thiol group le H is removed and it is displaced with the acetyl group, so that the SCOCH3. So this is now this is a peripheral arm. So the peripheral arm is now occupied with the acetyl group. Okay, so the H is removed in the form of CoA-SH. So this is the first priming reaction that is happening. Now we are having the first enzyme. The first enzyme we have found, that is, it is the monomer 1, whose acetyl transacylase works and it removes. What's the function of acetyl transacylase? It transfers the acetyl group from acetyl CoA, and it is kept on, or it is placed on the peripheral arm of the monomer 1, right? Peripheral arm of monomer 1. Okay, so that this is a peripheral arm. Peripheral arm of monomer 1 is now occupied with acetyl group. But one of the raw materials has come, right? Number 8 acetyl CoA or acetyl CoA will be coming in the form of acetyl CoA. Baki remaining 7 acetyl CoA will be undergoing carboxylation reaction. So the what is condensation, we should have 2 molecules, right? 2 molecules. So one molecule is now ready, right, for synthesis. Now we have got the other raw material. Other raw material known as second acetyl CoA. But the second acetyl CoA is coming in the form of malonyl CoA, right? So there comes a second reaction. So the malonyl CoA, we are having our malonyl CoA. The acetyl CoA has undergone carboxylation reaction. Now we are having the malonyl CoA with us. Now what is happening to the malonyl CoA? The malonyl CoA will be coming and getting attached onto the central thiol group, right? മറ്റേത് പെരിഫറൽ തയോൾ ഗ്രൂപ്പിലേക്കാണ് പോയത്, സിസ് എസ് എച്ച്. ആ സിസ് എസ് എച്ച് ഇസ് ഇൻ അസോസിയേഷൻ വിത്ത് ദ പെരിഫറൽ തയോൾ ഗ്രൂപ്പ് ഓഫ് ആരാണ്? ഓഫ് ദ മോണോമർ 2 ആണ്. മോണോമർ 2ന്റെ പെരിഫറൽ തയോൾ ഗ്രൂപ്പിലേക്കാണ് അയാൾക്ക് ഇന്ററാക്ഷൻ വരുന്നത്. അപ്പൊ അതുകൊണ്ട് തന്നെ ആ മെലനൈൽ ഗ്രൂപ്പിനെ ആരാണ് കൊണ്ടുവരിക? മോണോമർ 2ന്റെ മെലനൈൽ ട്രാൻസ്ഫറേസ്, റൈറ്റ്? സോ ദ മെലനൈൽ ട്രാൻസ്ഫറേസ് ഓർ the malonyl transacylase of E2 of monomer 2. E2 of ഈ പിക്ചറിൽ അങ്ങനെയല്ല കാണിച്ചിരിക്കുന്നത്. എന്റെ നോട്ട്സിനകത്ത് ഇറ്റ്സ് ഷോൺ വെരി ക്ലിയർലി. ഓക്കെ, ഇതിപ്പോ കാണാൻ മനസ്സിലാക്കാൻ എളുപ്പത്തിനാണ് ഈ ഒരു എംടിനെ ഇവിടെ ബ്ലൂ ഡാർക്കൻ ചെയ്ത് വെച്ചിരിക്കുന്നത്. ഓക്കെ, സോ ദിസ് ഇസ് നോട്ട് ബിലോങ് ടു ആക്ച്വലി ദ മോണോമർ 1. ദിസ് ഇസ് ദ മോണോമർ 2. ഇത് മോണോമർ 2 ആണെന്ന് മനസ്സിലാക്കണം. സോ ദ സെക്കൻഡ് എൻസൈം കംസ്, second enzyme E2 of monomer 2, that is malonyl transacylase of monomer 2, now comes into action. And malonyl transacylase, so what does it do? It picks up the malonyl group from malonyl CoA. The malonyl group, this is our malonyl group. Malonyl group is picked up from malonyl CoA and goes and gets attached onto the central thiol group, that is pan-SH, right? Pan-SH of the monomer 2. So that monomer 2 in the central thiol group, monomer 1 in the peripheral thiol group are now occupied with the 2 raw materials. Peripheral thiol group, we are having the acetyl group. The central thiol group is occupied by, our central arm is now occupied with malonyl group. Now we can carry out the condensation reaction. Malonyl group angane pona samayatha only a malonyl it now undergoes decarboxylation reaction. Once it gets attached, it immediately removes this carbon dioxide. So that net effect in the acetyl groups react. Clear? So the malonyl group malonyl CoA-SH is removed, right? Now the malonyl ACP, the central thiol group, CO, CH2, CH3, right? This carbon dioxide will be removed. Clear? So net effect, it is the two reactions, the